The overall goal of this procedure is to identify and surgically remove the adult zebrafish kidney, which can be used for applications such as cell isolation, transplantation, and expression studies of kidney and/or blood cell populations. This is accomplished by first selecting a 3 to 6 month old adult zebrafish for dissection. The animal is then sacrificed in 0.2% tricaine, with euthanasia being confirmed when the gills stop moving and the heart stops beating. The third step of the procedure is to use dissection scissors to open the abdominal cavity and to remove the organs located within it. The final step of the procedure is then to use fine dissection forceps to remove the kidney from the underlying connective tissues. Ultimately, gene expression profiles of hematopoietic or kidney cell subsets, isolation of specific cell populations through whole mount in situ hybridization, immunohistochemical analyses, and/or fluorescence-activated cell sorting can be performed on the isolated organ tissue and/or cells.
Visual demonstration of this method is critical, as the dissection steps are difficult to learn because the zebrafish kidney is a delicate organ that's anchored to the body wall via dense connective tissue.
The implications of this technique can be used in the therapy or diagnosis of kidney and blood diseases in humans, as biological processes are highly conserved between zebrafish and higher mammals. Numerous molecular genetic tools have been developed in recent years in the zebrafish, making it a useful animal model.
Pour enough 0.2% tricaine to cover an adult zebrafish between 3 to 6 months old into a dish. Then select a zebrafish for dissection and place it in the dish for approximately 5 minutes, watching carefully to see that the gills stop moving and the heart stops beating. Next, use a spoon to lift the fish out of the tricaine bath in a small amount of solution, decant the solution, and gently place the animal on a paper towel. Using a sharp pair of dissection scissors, make a cut just behind the gill operculum to remove the head of the animal. Discard the head in a biohazard waste container. Then, starting from the head, use the dissection scissors to make a long ventral incision that terminates at the tail to open the body of the animal. Now remove the internal organs of the animal using a pair of dissection tweezers and place them in the biohazard waste. Be careful not to brush the dorsal body wall, as this is the location of the kidney. If you have selected a female fish, scoop the developing eggs out of the body cavity with an angled pair of forceps or tweezers. Use dissection needles to pin the body walls to a dissection tray. Angle the pins so that the kidney can be visualized. The kidney possesses a characteristic shape with a head, a trunk or saddle, a tail, and a characteristic colour, being interspersed with black-hued pigmentation. Use 2 pairs of fine forceps to detach the kidney from the dorsal body wall. Use 1 pair to lift the kidney, and 1 pair to hold the underlying connective tissue as you remove the organ from the zebrafish body cavity. Gently place the kidney into the desired vehicle for further live cell studies, such as a microcentrifuge tube containing 1 times PBS for fax preparation. Fill a dissection tray with enough freshly prepared fixation solution to submerge a 3 to 6 month old zebrafish during a dissection. Then pour enough 0.02% tricaine pH 7 into a dish to cover the zebrafish. Select a zebrafish for dissection and place it into the dish of tricaine for euthanasia. Remove and discard the head of the animal as shown previously, but this time immediately place the animal into the PFA containing dissection tray after the removal. Keeping the body partially submerged in the fixative during the procedure, use dissection scissors to open the body of the animal with a ventral incision, as before. Again, being careful not to brush the dorsal body wall, remove the internal organs of the animal using a pair of dissection tweezers. place the organs in a biohazard waste container. As before, use dissection needles to pin open the body walls, angling the pins so that the kidney can be visualized. Then fix the sample overnight, placing the tray at 4 degrees Celsius. The next day, remove the fixative solution from the tray and replace with 1 times PBST. Then, as before, carefully detach the kidney from the dorsal body wall, using 1 pair of forceps to lift the entire kidney organ in one piece as you separate it from the connective tissues that underlie it. Next, use a wide-bore transfer pipette to place the kidney into a microcentrifuge tube for further processing. As shown here, following the removal of organs in the body cavity, the kidney appears as a single flattened organ that is adherent to the dorsal body wall via connective tissues. In this figure, the kidney has been schematized to show its characteristic anatomical shape. Here, cadherin 17 transcripts, detected by whole mount in situ hybridization of the kidney, can be seen in the tubule of adult kidney nephrons, with an enlarged view of the tubule staining shown in the right panel. In this figure, it can be seen in the left panel that MAFBA transcripts are localized to proximal nephron cell types in the adult kidney. The enlarged view of MAFBA expression, as visualized on the right, reveals that the transcripts are specifically localized to the podocyte cells of the glomerulus, as indicated by the P and arrow, and to the proximal convoluted tubule, indicated by the PCT and bracket.
While attempting this procedure, it's important to remember to remove the internal organs of the adult zebrafish with care, so not to damage the kidney prior to its surgical isolation.
Following this procedure, other methods such as histology or fluorescence-activated cell sorting can be used to assess the ultrastructural characteristics of blood and kidney cell populations, or to isolate specific populations for cell transplantation or primary cell culture.